DNA is a nucleic acid that contains the genetic information of cells. It basically gives the instructions for functioning and developing of all organisms. An easy way to understand DNA is to compare it to a blueprint. Just like a blueprint, it gives the instructions needed to construct a cell. Protein synthesis is how cells make protein. The first key to protein synthesis is transcription. Transcription is when RNA copies information down from a DNA strand. The information that is copied down is known as RNA. Messenger RNA, also known as mRNA, takes it to a ribosome. The next step in this process is known as translation. The mRNA is read and used. A polypeptide chain builds up. When this chain is complete, it folds, forming protein. The cell cycle is a chain of events that is key to cell division. The cell cycle is composed of 3 cycles: interphase, mitosis, and cytokinesis. Interphase is when the cell grows and matures. The stages in interphase are characterized by growth and development. During this phase, DNA in the cell is duplicated. Prophase is the first phase of mitosis. Early in this stage, the cell membrane disappears. Chromatin condenses from chromosome spindle fibers. At the end, the nuclear membrane dissolves away. The next stage of mitosis is metaphase. Chromosomes are attached to spindle fibers which change in length, pulling chromosomes into a line along the equator of the cell. The third phase is anaphase. The centromeres split and the sister chromatids are pulled to opposite poles of the cell by the spindle fibers. When the chromatids reach the poles and stop moving, the phase is over. The final stage of mitosis is telophase. The nuclear membrane forms around each set of chromatids and the nucleolus reappears in the nucleus. Then the chromatids unwind, forming chromatin. Cytokinesis is the stage when the sides of the cell pinch inward, dividing the cytoplasm and forming 2 daughter cells. Both of these cells have a complete copy of their parent cell's DNA. Cancer starts when a group of cells lose contact with the other cells in the body. Cell communication is important to control numbers. So when that group of cells loses contact, they start multiplying out of control. As the abnormal cells multiply, they never get a signal to die. Cancer cells have receptors that take in a nutrient known as folate. This is where our solution begins. This is my nanobot. It is based off the idea of a different source of nanotechnology, carbon nanotubes. Nanotubes are half the width of a DNA strand, so many could fit in one cell. When a carbon nanotube is under near-infrared light, the electrons get excited and they give off heat. My nanobot is very similar to nanotubes. It starts with an injection of millions of our nanobots. After the injection, my nanobot will be inside the tumor. Now the nanobots need to find cancer cells. Just like the nanotubes, my nanobot is made of carbon. My nanobot is just slightly smaller than a nanotube. So what will happen after the nanobots are injected into the tumor? Because they carry that nutrient folate, when they pass by normal cells, they won't be taken in. When they pass by a cancerous cell, the folate receptors will take in the nanobot, thinking it is folate. Once all the nanobots are in position, the doctor will simply zap the skin above the tumor with near-infrared light. And just as we talked about, the nanobot will give off heat, thus killing the cell. With this kind of solution, it can save months of patients suffering through chemotherapy and radiation. More importantly, it could save lives.